All, this is Dr. M. Arumugam, Assistant Professor and Head, Department of Botany, J.J. College of Arts and Science, Pudukkottai, Tamil Nadu, India. In this presentation, I would like to say something about the somatic hybridization. What is somatic hybridization is the development of hybrid plants through the fusion of two different plants or plant species or varieties protoplast is called as somatic hybridization. In the picture we can see A plant is a different plant and B plant is a different plant, so we are going to merge together A and B, so we are derived A and B to AB plants. So in AB plants we can have the two different characters from A plant as well as the B plant. So the merging or joining of two desired characters from two desired plants. Next, what is protoplast means? The protoplast or the cell by removal of cell wall. Usually it is done by digestion of some enzymes or also some mechanical methods. Some enzymes used to say the cellulose enzyme digests the cellulose in the plant cells, while the pectinase enzymes break down of the pectin holding of two individual cells. Once the cell wall has been removed, the resulting protoplast is a spherical in shape. And what are the applications of the protoplast culture or protoplast means? In this, we can have the protection of a hybrid for somatic hybridization, overcome the sexually incompatible species, ingesting the foreign material into the cytoplasm, and for a DNA transformation used to study the wall synthesis as well as the decomposition, and studied as the techniques of a single cell system. And somatic hybridization is in the slide we are giving a generalized flow chart. And first step we have to isolate the protoplast, and fusion of protoplast in a desired species or varieties, identify and also the selection of the somatic hybrid cells and culturing of somatic hybrid cells, and in final we have to regenerate the hybrid, that is somatic hybridization techniques done in the plants. And next slide, first step is isolation of protoplast. The first step is isolation of the protoplast, and it is done by two different methods, one is a mechanical method as well as in a enzymatic method. And in mechanical method of isolation of the protoplast is done by the, we have to take these plant tissues, and this plant tissue is allowed to cell plasmolysis. After cell plasmolysis we have to observe the cells by the microscopic observation, and we have to cut the cell with a sharp knife of cell wall of the cell, and we have to release the protoplasm. Then we have to collect the protoplasm accordingly. Then the mechanical method is done. In this illustration we can see the tissue is immersed with 1.0 molar sucrose until the protoplasm shrinkage away from their enclosing of the cell wall. So when you attain the plasmolysis, then the cell has to cut by using a very sharpened plates of the cell wall. Then it is used to vacuolate the cell like onion bulb, scales, radish, and also beetroot tissues also. In this method of mechanical method, we can yield or we can take the very low quantity of the protoplast, and we need the skilled person and tedious process also. Low protoplast viability will be taken or will be observed in the method of mechanical method. And next method is enzymatic method. The method is it is quite common as well as it is a recommendable method. Leaf sterilization, removal of epidermis. First step is we have to sterilize the leaf as well as we have to remove the epidermis of the cell leaf. Then we can have the two steps or two methods of enzymatic method. One is we can allow to put in a plasmolyzed cell with the two enzymes, pectinase as well as the cellulase, and we can have the protoplast released, and we have the isolate the protoplasm. And another one of the method is we have to treat the cells with the plasmalized cells with the two steps. First we have to allow the pectinase, and we can get the released isolated cells. The pectinase is used to isolate the cells from the joining of two cells. And another one and followed by the cellulose is applied for the protoplasm isolation. Now we have taken the isolation protoplasm, we can take the protoplast isolation. The next enzymatic method, we can this slide we can see shoot culture and the removal of we can have the leaves, and we have to cut into the leaves in a very small pieces. Then we have to incubate the enzymes and isolate the pectinase, and we have to allow for the centrifugation, and we have to collect the isolated protoplast. In the method is used variety of tissues, organs including the leaves, petioles, fruits, leaves, coleoptile regions and hypocotyls, stem, shoot, apexes, and embryos also we can have some microspores also we can have in this method. And the most probably the mesophyll cells is suitable for the isolation of the enzymatic method of protoplasm. And in this method we can have the high yield of protoplasm and easy to perform and more protoplastic viable. So isolated protoplasm will be more viable in this method. The enzymes which are used to break the cell wall is various enzymes used for this process. First is cellulose and pectinase, or otherwise mesozymes for the plant cell wall, and bacterial cell wall is degraded by the lysozymes, and fungal cell wall will be degraded by the novozymes 2, 3, 4, and otherwise called as glucanase and chitinase, because you know that the fungal cell wall is made up of chitinase. So the chitin, so the chitin will be degenerated with the chitinase. The septomyces cell wall degraded by the lysozymes and achromo-50 and this now we have isolated the protoplast. We have to get purify how much it is pure of the protoplast we have derived, means we have to check it over. Then for this we have to have the protoplast to purified by the removal of undigested material, that is debris and burst. Then is broken protoplast has to be removed, and we have to remove the enzymes which are employed for the isolation. The debris are removed by filtering of preparation through the nylon meshes, and enzymes will be removed by the centrifugation where the protoplast is settled at the bottom, and the tube will be collected in the supernatant, and intact protoplast are separated by the broken protoplast through the centrifugation and removed by the pipettes and using the collected of the stops. And in the next step is the protoplasm fusion. This method will be followed by one is two types. One is spontaneous fusion as well as induced fusion, and the spontaneous fusion is intraspecific and intergeneric, and induced fusion will be have by the hemofusion, mechanical fusion and electrical fusion, that is electrofusion also we can have. In this method the protoplasm fusion is classified into two categories. One is spontaneous as well as induced. In the first step, often the fuse is spontaneously fused through the plasma desmata, and the induced method we should have add some fusion chemicals is called agents, normally do not fuse with each other. Then how the isolated protoplast can be induced to fuse means, first is hemofusion. Several chemicals have been used to induce a protoplasmic fusion, such as sodium nitrate, polyethylene glycol, calcium, and also calcium ions, calcium 2+ ions also will be employed. And the cause of the isolated protoplast to adhere each other, now it is non-specific, it is inexpensive. It can use the massive fusion approach. It can be cytotoxic and non-selective and having the less fusion of frequency. And the electrofusion is nothing but by using of electric stimulation in the field of protoplasmic region where an electric field will be low strength. The electric fusion is to control easy to control, it is having the fusion of frequency of the 100%, it is give reproductibility also, it is less cytotoxic, and the last one is the equipment is sophisticated and also somewhat expensive. This slide we can have the illustration of our mechanism of the protoplasmic fusion. The first one is we have taken the two different varieties, isolated protoplasm, then we have harvested cells and count apical desired genes, so we have to merge with one another with the fusion chamber, then we have to allow for this electrical field for the electrical fusion, then the two different protoplasm will be fused together, then we have the hybrid protoplasm. The protoplasm in a bacteria, the protoplast can be obtained, fuse can be carried out with a low frequency of somewhat positive organisms. For gram negative bacteria, it is possible to obtain the protoplast, but regeneration is difficult. The procedure is highly efficient and yields up to 80% of transplants. Now how to culture, now we have taken the somatic hybrid protoplast, so we have to culture the protoplast. So isolated protoplast can be cultured in appropriate medium, and that is called reform cells, and as well as regenerate the calories. So because we have to regenerate the cell wall, then only we have some structure, then only we have the protective purpose, then only it will have some mode of actions also. So the optimal culture conditions, the optimal density to the culture, we have to maintain it. And second one is optimal oxygens to cytokinin ratio, glucose and sucrose also we have to be added. Maintain the osmoprotectant in the medium, and we have to allow the temperature for 28 degrees Celsius. The pH has to be maintained 5 to 6, 5.5 to 6 we can have. So 0.25% calcium hydrolysis also we have to be added. Then what is called BAP, the benzyl adenopurine, as well as naphthoacetic acid, the hormones are to be added. In a culture of protoplast, the protoplast cultured in a suitable nutrient medium, with the first we have to generate the new cell wall, and another one is the formation of a complete cell with the wall is formed by an increase in size, numbers of organelles, and induction of the cell divisions. The first cell division may occur within the 2 to 7 days of culture, then resulting in the small clumps of the cell, also known as microcolony, within 5 to 3 weeks. And from such clumps, there are two routes to generate the complete plants depending on the species. The plants are regenerated through the organogenesis, like a callus, and from the callus we can have some shoots, and otherwise whatever the need we can do it by the callus. And another one is microcalli can be made to develop the somatic embryos. So from the callus we can produce a somatic hybrid embryos. From the embryos we can have some whole plant from the regenerations. What are the advantages of the somatic hybridization is we can produce the novel interspecific as well as intergeneric hybrids also. For an example, potato hybrid of two potato and tomato, we can have the potato. Then in production of the fertile diploid and polyploid from the sexually sterile haploid, triploid and uneuploid plants also. Now we can transfer the gene from the disease resistant, abiotic stress resistant, herbicide resistant, and many other quality characters also we can have to transfer from one to another. We can have a good variety also. Then production of heterozygous lines in the single species which cannot be propagated by the vegetative means. So some of the plants we cannot be produced by vegetatively. For other plants we can go for the somatic hybridization. The studies of the fate of the plasma genes. So we can have the fate of the plasma genes. We study what are the mechanisms over there, how we can modify it, then we can do that over there. The production of the unique hybrids and nuclease and also cytoplasms. And what are the limitations of the somatic hybridization means? We have the very poor regeneration of the hybrid plants. So we can have the isolation and hybridization and also culture of the protoplast. In the case of regeneration from the somatic hybrid done, the somatic is very much tough also. Non-viability of fused product. So when you fused two isolated protoplasts, it is sometimes it also non-viable things also, not successful in all the plants. So we can do some certain plants, but in all the plants it is difficult to success. The protection of the unfavorable hybrids, lack of an efficient method for selection of hybrids, no confirmation of the expression of particular traits in the some of the hybrids also. So in next one is, what are the factors which influencing the culture of protoplast culture? One is plant species and varieties. So it depends upon the plant varieties and species also the somatic hybridization is done. Some of the genetic difference leads to varying the protoplast responsible to culture the conditions. And the second one is plant age and also organ, how far it is old, then what organ we have taken for the somatic hybridization. So in the case we have selection of the explant like leaf is or petiole is good, but instead of the other organs also. The pre-culture conditions, the protoplast culture are highly influenced by the climatic factors, and different cultures of the seedlings yield protoplasm have the different responsibility when cultured in the somatic hybridization techniques. Pre-treatment to the hybrid, the isolating the protoplast. So cold treatment, plasma lysis, and hormone increases, and chance to recover of the viable products protoplast, and although their planting of efficiency is very much difficult also. The density, the crucial as it influences the plating efficiency and surviving of the protoplast at in a high density. Protoplasm complete with one another, while at a low density losses of the metabolites from the protoplasm more. Thank you for watching. Thank you very much.